Episode 16 of the WildFed Podcast, The Truth About Bluefin Tuna with Molly Lutcavage, is brought to you by Surthrival. This Valentine's Day, skip the chocolates and roses and instead indulge in one of Surthrival's premium love tonics. Surthrival's legendary elk antler velvet is a tonic for men and women that's long been revered for its potent adaptogenic and regenerative properties. This nutrient-dense elixir contains a powerful blend of growth factors that enliven inner drive, stamina, and endurance, as well as help to balance testosterone and other sex hormones. Surthrival's Taboo is an herbal and animal tonic that was originally formulated for women, but guys, I'll be honest, it's one of my favorite products in the line, and I love to take a double dose before I head into the bedroom. It contains a suite of aphrodisiac and hormone-supporting herbs and a base of elk antler velvet and a heavy dark chocolate. Adult women of any age will benefit from its libido-enhancing qualities, and women over 40 will find targeted hormone-balancing support. But again, guys, don't skip this one. It functions like an herbal Viagra. Now through Valentine's Day weekend, save big on all Surthrival elk antler velvet, Taboo, Shizandra, and CBD3 products too. Shop now at surthrival.com. For those of you that ordered Season 1 of the WildFed TV show, you received Episode 6 yesterday. That episode's called A Heavy Harvest, and it's all about the most calorie-dense wild food harvest of our year. First, Avani and I head out into the wild rice stands by canoe, along with Arthur Haynes and his wife Sarah. Arthur teaches us about wild rice as a plant, and then I take our harvest home and walk you through processing it into one of the most sought-after wild foods in North America. This episode also follows Arthur and I, along with a couple of houndsman friends and their hunting dogs, on 2 black bear hunts. In addition to the meat, you'll see how I render bear fat into a clean and healthy cooking oil. We also harvest some delicious culinary mushrooms and then bring those ingredients to Chef Evan Hennessy for a celebratory meal at his restaurant Stages in New Hampshire. I love the cinematography and storytelling in this episode, and so it's probably one of my favorites of the season. If you aren't currently subscribed to the show, go over to wild-fed.com to purchase Season 1. 6 episodes are available to watch today, and you'll receive a new episode each Monday until all 8 episodes have been released. One more bit of housekeeping before today's episode. Please, if you're receiving value by tuning into this podcast, let us and the hosting platforms know by going over to iTunes and leaving us a 5-star rating and a personalized review. Making this show takes a ton of time, money, and effort, but you leaving a review only takes a moment. Our ability to get great guests and grow our community depends heavily on our rankings, and you can help us do that. If you're one of the folks who've already left an incredible review, I want to personally thank you. You are appreciated. I'm Daniel Vitalis, and you're listening to the WildFed Podcast, a show about reconnecting with nature through hunting, fishing, foraging, and food. WildFed. Food is all around you. Okay, today's show is one I've been eager to release for a while now. As someone with a passion for fish, fisheries, and eating wild seafood, there's probably no species consumed as controversially here as bluefin tuna. They're big, huge in fact, some well in excess of 1,000 pounds, and they're charismatic, so they get a lot of media attention. From shows like Wicked Tuna to the near constant news stories warning us about the stock declines and the near threatened status of the species due to overfishing. With some groups like the IUCN even listing them as endangered, we've been inculcated with a mixed tuna message. On the one hand, we see it in nearly every sushi restaurant in the world, since maguru and toro are nearly ubiquitous options on sushi menus. Toro, by the way, is the fatty belly meat of bluefin, and if you've ever had the real thing, it's probably one of the most divine foods you've ever had melt in your mouth. It's basically raw, uncured tuna bacon. But I digress. While we see market forces driving the commercial fleet to continue fishing for this species, we see simultaneously regulatory pressures and outcry from environmental groups that are slowly working to shut the fishery down. I've always felt conflicted, since I both love to eat Atlantic bluefin and hope to one day fish for them. But of course, I'd rather see them left in the sea if they are truly endangered, or even just critically threatened. But what if the truth wasn't so simple? What if there is a discrepancy between what the scientific evidence tells us about Atlantic bluefin stocks and what the predictive models are telling regulators? Today's guest is no stranger to the controversies that swirl around Atlantic bluefin. Dr. Molly Lutcavage is an oceanographer and a tuna and sea turtle physiologist. She's the director of the Large Pelagics Research Center at the University of Massachusetts, Boston. She's had an exceptional career conducting research across the world's oceans and believes, based on field observations and newly discovered physical evidence, that the Atlantic bluefin might not be imperiled at all, but may in fact be thriving. This belief, which stands in the face of the standard bluefin narrative espoused by fisheries managers and parroted by environmental groups, has led to a significant loss of her research funding and attempts to smear her professional reputation. But, a true scientist, she continues to follow the data and spread the word. Atlantic bluefin may be doing better than you've heard. I think you'll find this interview to be fascinating and Molly to be a brilliant, passionate, but grounded scientist with an incredible story to tell. Should you be eating Atlantic bluefin tuna? Give this interview a listen and then decide for yourself. I might just be seeing you at the sushi bar. Hey, I'll be back next Tuesday when my guest will be Catherine Hopkins, professor at the University of Maine's Cooperative Extension and director of the International Maple Syrup Institute. She'll be here to teach us all about one of my favorite wild foods and probably one of yours too, maple syrup. Until then, get ready to do a deep dive and a long migration into the world of the Atlantic bluefin tuna. Well, I am here in Gloucester, Massachusetts with Molly Lutcavage. Lutcavage, you say it for me.
Lutcavage, good right. Ukrainian, Lithuanian.
Oh, is it?
Name
okay.
from Pennsylvania.
I'm really excited to be sitting down with you today. I've been reading articles of yours, watching videos, reading some editorial stuff, and your work on bluefin tuna has really fascinated me. So I'm really grateful first just for the opportunity to be here. Can you give us a little bit of perspective? I want to talk about your credentials in a second, but first give us a little perspective on this place and what role it's played historically, because I feel like from the perspective of fisheries and seafood, we're in a really historic part of the United States right now.
You're right. You're sitting in East Gloucester, and just down the street on East Main Street is one of the last standing cod flake facilities where the fishermen laid out their cod to dry.
Oh, no kidding.
And, for the salt cod, but it was the dried cod, and that's at a marina just down the street is still there. So, yeah, clearly Gloucester has been, the historical point of our fisheries for first inshore fisheries, Stellwagen Bank, and then eventually George's Bank. The schooners were built nearby in Essex, which is just, a few miles west of here. And, of course, we have the very famous captains that ran the clipper ships, and we have Charles Birdseye. Birdseye is credited with inventing and having the first
the freezing
fish,
right?
plant,
Yeah.
which was just
Birdseye. People are thinking
right.
maybe of the frozen fish company, and that's why.
That's it.
Is it actually? You may not know this, but is that still a family lineal thing, or is it just the name has stuck in the current incarnation of that company? Do you know?
It's the companies are now international, and in fact,
right.
the Birdseye, the original building, which is an icon in the landscape of Gloucester, was torn down. The new hotel is sitting where the Birdseye, the famous Birdseye plant was. But so Gloucester has also been the location for incredible ocean innovation. From not just the designs of the ships that were fishing, but as you know, Charles Birdseye, the processing. Herb Holton, who is out at the marine station that we occupied for a bit. Herb Holton is a famous fish processing, food, managing basically how to better prepare fish and store fish.
Okay.
So he's very well known. Sadly, he's passed on now. But so Gloucester still remains a point for centuries now of innovation in seafood, and not just seafood, but the ocean, and basically getting to it to and fro safely.
Okay.
And as you can see, our harbor is greatly downsized, and it's obvious, same as up and down the coast in Portland and New Bedford, just the impact of the changes in our ocean and fisheries management and so on and so forth.
Sure. Was it cod that was the main fishery here for a long time?
Not just cod, redfish, the cycles of the coming and going of the different
Was
targets.
there a big commercial fishery for redfish here?
Absolutely.
We're talking redfish, the very old, small, big bug-eyed. I don't know what the genus is of that fish.
Oh, I forget. I should know this. I'm a tuna specialist, but I'm rusty. But and I mean, it's interesting.
I catch them sometimes ground fishing, but I don't see too many of them, and I didn't realize there was ever any significant fishery up here.
Oh, there was, and in fact, the tuna seiners that are all now gone, but the tuna purse seiners would, before they fished for bluefin, they would go back and forth between herring, mackerel, and redfish.
Oh, okay.
So, which, that history is not well known. I know it because the Genovese family, who I fished with for 7 years on their purse seiner, doing, scientific work, they had that whole history of the different kinds of fish that they targeted depending on, good years and bad years.
Right. And then how about academically? Is Gloucester played a role, I guess, in, I don't know how to say it, ichthyology, oceanography?
It did.
All of that.
Not per se from the university, but the Bureau of Commercial Fisheries started in Gloucester. And so that has become National Marine Fisheries Service. So
that's federal.
Federal. So the Bureau of Commercial Fisheries did the first exploratory longline cruises for tunas in the Atlantic. And a lot of the original work back when National Marine Fisheries, their job was to help expand our U.S. fisheries and make them economically viable and productive.
What era are we talking about here?
I don't know when they started, but probably in the century, there were forms of fisheries science and management, but I know that it was Bureau of Commercial Fisheries through at least the 60s, because the exploratory longline cruises that were out of Gloucester, they were longliners, so scientists got on longliners, commercial longliners, and did the work. And there weren't research vessels, so they always worked with fishermen. Yeah. So in the early days, the old-timers meaning the first fisheries scientists in Gloucester, were very much tied into our fisheries. They were neighbors, they were buddies, so they did field work, field ecology on the high seas, which has been my lab's model from the early days, because the basics of fish life history, what you eat, where you spawn, how long you spawn, all of that. The old timers in fishery science and the Bureau of Commercial Fisheries, that's the work they did. It was way before mathematical models and statistical models for assessment. They were doing basic biology, and a lot of that was done, on the commercial boats in Gloucester.
Yeah, which makes a lot of sense where you have that fleet going out. Tell me a little bit about your academic background and then the work that you've done in your career. You have a pretty illustrious background from what I can tell.
Yeah, an infamous background in. I'm trained as a biological oceanographer. I did my first work was at William and Mary College, Virginia Institute of Marine Science, and honestly, I got hooked early on with the ocean as a kid growing up in the mountains of Pennsylvania, and I was born in '55, so I'm a TV kid that watched Sea Hunt with Mike Nelson, underwater detectives, and then there was a show called Adventures in Paradise, and that was taking place supposedly in Polynesia on a schooner with diving. And so I became fascinated with diving in the ocean.
But living inland.
Living upstate in the Poconos. Yeah. And I don't know, I guess I was hooked early on, like a lot of people. And if you ask a lot of people my age that are oceanographers, almost everyone says that Sea Hunt and Jacques Cousteau and Adventures in Paradise influenced their career. So anyway, I was fascinated with diving. I was a diver in undergrad, and I just loved being scuba diving and also diving physiology. I wanted to become an MD that studied diving physiology of humans.
Right.
My undergrad,
which is a fascinating area of research.
That's right, and it's a man. So I was told by my undergrad mentor, I worked in a hyperbaric center that was, pushing the deepest dives ever for humans at the University of Pennsylvania, where I did my undergrad.
These were free divers, or these were scuba divers?
These were commercial divers in a hyperbaric research setting, so in tanks. But anyway, my boss basically said, a woman will never be allowed on any rig. And so just forget about it.
What year was that?
That was in 76, 77.
That's changed.
I had to shift, and I took a course in Maine, Swans Island Marine Station that Penn owned, and that was it. I got out of pre-med, and I just decided I needed to study the ocean. But diving was still there. I couldn't study humans anymore, in theory. So what's left, marine mammals and sea turtles. So long story short is that I became a sea turtle diving physiologist and studied diving, breath hold diving in marine sea turtles, diving birds, and of course mammals.
Oh man, that's like a whole nother topic I wish we had time to go into. I'm really fascinated by that, and that whole mammalian dive reflex. And we just yesterday were sitting down with a friend of mine who's a free diver out of Portland, Maine, spear fisherman. But there's a community of maybe 10 people there who do that. But we were talking a little bit about the mammalian dive reflex, and just very fascinating physiologically. But you went on to get a PhD in what's your
diving physiology?
That is what it's in.
Yeah, I mean, it's really about how animals work, and so we're in a group of people that were known as comparative physiologists or physiological ecologists, and that is, you don't just study one system. It's what are the commonalities and the differences. But breath holders in the ocean, they're not fish. They're the ones that have to be really good at figuring out how to get down there and get their food and everything else, whether you're a mammal or a seabird or a sea turtle. So that's what got me into it. But the PhD was looking at diving physiology and free diving sea turtles, and that got me into technology, using transmitters and so on. But that also got me to bluefin tuna, because the connection from my training and my own personal passions in science were animals on the edge of the extreme that were really good at what they do. Leatherback turtles are the only living warm-bodied reptile that are left.
I saw in something I was reading about you that this connection of bluefin being a sort of a warm-blooded fish as well as the leatherback turtle, and I had read that a lot about bluefin. I'd never heard that about turtle. Could you kind of give our audience like a little bit of I don't want to spend too much time on this, but give us like overview briefing on the status of sea turtles and whatever you want to share about leatherback turtles, because it's fascinating. I just saw, I hadn't really run into them much, but I saw a big one offshore this year.
A leatherback.
Yeah, off the coast of Ogunquit. Maybe we were probably on Jeffries.
There are so many commonalities between leatherback turtles and bluefin tuna.
No way.
And really? New Englanders, some of the old timers know this. So why? They're both here. They're both here because they can maintain their body temperatures much warmer than the ocean that they're in. And bluefin, besides poor beagle sharks or laminate sharks, they're the only living fish that keeps a body temperature much higher, just like leatherbacks. What that means is they have almost no restrictions. I mean, literally they have, but they can be found from 32 degrees Fahrenheit all the way up to 86 degrees. They go from the surface. Both leatherbacks and bluefin can dive on a regular basis if they want to 3,500 feet. So in other
I would
words,
not have thought of turtles going that deep.
Leatherbacks.
Okay.
And the other thing is about leatherbacks, like bluefin tuna, they pack, they soup up their red mussels. And so they have really interesting adaptations that allow them to do things that no other members of their, groups like the tunas, big eye can do some cool things, but they're not bluefin. Leatherback turtles are the only living member now of what were like the warm-bodied dinosaurs. They grow fast. They have mammalian bone growth types. They're different, and they're the only ones left. All the other members of those kinds of reptiles, dinosaurs, and sea turtles are extinct.
Yeah.
So
what's their conservation status now?
In the Atlantic, leatherbacks are doing pretty well.
Okay.
They, there were lots of threats to their survival. The Pacific is still a real problem. Climate change, development. I mean, not just fisheries. Fisheries get blamed for everything, but habitat and loss of habitat and everything else. The Atlantic leatherbacks are actually expanding their nesting ranges, they're I think conservation efforts of the last 30, 40 years, at least as long as I've been involved, have changed everything particularly for Atlantic sea turtles. The Pacific is much more challenging, and it's a bigger ocean, and there's a lot more variability in the productivity regimes, and so the Atlantic's kind of small in comparison with the Pacific, and so...
Is the leatherback species here in the Atlantic the same species as the Pacific, or are they different species?
They're the same species, but they have different... they're like different tribes. They have slightly different physical characteristics. The Atlantic leatherbacks, I hope I get this right, my memory's going on, they get bigger. They're bigger, final body size than Pacific. they have slight differences in their life histories, but they're the same species.
Okay. How did you come to be involved with bluefin tuna? From turtles?
Yeah. Well, in Vancouver, my lab, the diving physiology lab at UBC, was studying different diving ducks.
Okay, so you were up in BC.
I was up in BC for a postdoc at the University of British Columbia and still studying diving animals. My lab was a diving duck lab. I got them into the leatherback work. But at the same time, my lab, my boss, David R. Jones, who was a famous, diving physiologist, he'd say, "Come on, Mala, you don't want to." He called me a turtle hugger. He sort of he didn't know anything about sea turtles, but he became obsessed with leatherbacks because they're such a cool animal. But my lab, Dave Jones's lab, was studying bluefin blood.
Oh, okay.
And because he studied the oxygen binding, because we're diving physiology, the oxygen binding characteristics of the blood, basically, it's like your scuba tank. How much blood or oxygen can you store? And so diving mammals...
They use hemoglobin like us?
They use hemoglobin and myoglobin. So
that's in the muscle.
In the muscle, you're right. So mammals, like the whales and stuff, seals, they load up their oxygen in their muscle in myoglobin.
Okay. We don't do that as humans?
Not as much. We have much less. We have a lung store.
Okay, so when we dive, we gotta load up our hemoglobin in our blood with oxygen, but these animals that are more adapted to marine environments are able to do that in the muscle as well.
That's right, and that's why they're dark red meat, as you know. And so, coming back to the tricks, leatherbacks are pretty much the sea turtle that has myoglobin levels comparable to mammals.
Wow.
So they can dive deep because they load their oxygen in their muscle, but their lungs are small compared with other sea turtles because they have to be small because they don't want to have the bends, so they don't have the big lungs that shallow diving turtles have.
Okay.
So they're sort of functioning like a marine mammal.
That reduces the amount of nitrogen they bring down with them, essentially.
That's right. Their lungs collapse, and they're more or less a functional, I sort of say it lightly, marine mammal.
Okay.
They keep their body warm. They have counter-current exchange, just like a bluefin tuna, to keep the cold, the extremity blood away from their core.
Right.
So bluefin tuna and leatherbacks have a whole kind of similar tricks.
It's convergent evolution.
Absolutely convergent,
from
adapting.
2 different lineages. They're at, how deep did you say?
We speak in meters because we're scientists, but let's say 3,500 feet seawater. So 1,200 meters.
There can't be
for
much
both.
light or any light there.
There isn't, but what leatherbacks have big eyes, and like big-eyed tuna and tunas. Leatherbacks in particular, as you know, they're queuing in on gelatinous. They're specialists. They eat jellies. And that's 80%. Edie Whitty, who is she's basically one of the leaders in the phosphorescent, luminescent deep ocean.
Oh, yeah.
Her lab found that about 80% of the deep sea are luminescent.
So they can see their prey when they get
absolutely,
down there.
And they have big eyes, and leatherback eyes are adapted for light gathering. So it's all about physiology.
That's so fascinating. My goodness.
It's really cool. I mean, this is the sort of stuff you don't hear about in stock assessment and fisheries science, but this is how animals work. And for me, that's the science I love.
Oh, it's so exciting. So you're there looking at tuna blood.
My boss was. I wasn't. But I knew about bluefin because they were like leatherbacks. So the segue from my academic career to postdocs in diving, I took a federal job in the Gulf of Mexico, and I hated it because I was not doing science. And so I quit. And I was still interested in leatherbacks in New England. I called my friend and a colleague at the New England Aquarium that had a database on leatherbacks that they had when they were flying surveys. I said, "Hey, how about can I see your leatherback aerial survey data?" And he said, "Sure, you can have it, but what are you doing right now?" And I said, "Well, I'm kind of in between opportunities," sort of jokingly. And he said, "How would you like to run a bluefin tuna aerial survey?"
Oh, cool.
And I said, "I don't know squat about bluefin tuna aerial surveys, but I've done sea turtle surveys, so sure, why not? They're warm-bodied." So I got hired on a short contract to run, that was the spotter pilot survey for Scott Krause, who became my collaborator, and he was my boss, and that was the beginning of the Bluefin Tuna research program at the New England Aquarium.
What year was that?
93.
What was the one of the things we're going to be talking about today is the sort of landscape of tuna conservation and fisheries. And it's a much more contentious place than people might realize who only see occasional headlines and don't pay a lot of attention to fisheries, or in particular to tuna. And so I'm excited to look at sort of some of these contentious issues. But what was the landscape like in 1993? What was sort of the consensus about bluefin, the fishery, about the... And actually, can we back up a little bit? I just was talking to Grant on the way down here, and everybody, Grant, our producer, is sitting here silently in the background, but is with us today. And on the drive down here from Maine, I was telling him I don't have a lot of personal experience with bluefin, but I've, in the last year, for whatever reason, been reading a lot of books, watching documentaries, kind of listening to anything I can find. I've been obsessing a little bit. I saw a nice bluefin come out of Pine Point in Scarborough, and it just humbled me when I saw this fish come out of the fish hold. I just needed to learn everything I could. So
wow.
The story that I have encountered is that when the fisheries began here in the Northeast, it was sport fishermen. They didn't even see a food value, and that they would kill them, bury them in the sand and let them rot. And then eventually they went into the cat food market. And then it wasn't until the Japanese kind of came over here. And I also had come to understand something about maybe the Japanese didn't traditionally necessarily eat a lot of red meat tuna, but that their sort of Western influence of steak eating had influenced the sushi market, and that this boom happened. But it's not, bluefin tuna, at least in the Northeast, is not a long-term traditional fishery, correct? I don't know if there's a question in there.
Yes and no. And to the no part of that, I became aware of with my colleagues when I was at UNH, and there was a historical fisheries group there, and I had pictures that they supplied to me, and bluefin tuna stacked up with swordfish in a fish house in Maine. And what's really interesting is
Going back how far?
1900.
Oh, wow. Okay.
And so what was interesting is there must have been, what's the word, artisanal or local take that I don't know a whole lot about that knowledge, but it's certainly there, and the pictures show that it's true. But what was interesting is before the damming of rivers, and as you know, the salmon, this is your area, apparently swordfish were coming all the way up into the rivers.
Oh, no way.
And
really? I've never heard that.
Yep, and that they basically didn't have to go offshore to catch some of the pelagics.
They would have stayed below the head of tide. They're not anadromous or anything, right?
No, they're not. But there they were on the dock. The pictures show that it was true. Now, I looked back into the Bureau of Commercial Fisheries and the state fisheries records for what they were catching here, and I didn't see tuna listed in the state of Massachusetts, but clearly they were being landed in Maine.
Okay.
And so coming back to, you're right about the commercial history, but the other thing was that there's two thoughts that I had about that. So yes, you might someday follow up on who was catching those.
Right. I wonder too if there was an indigenous harvest.
I was going to say that it's an interesting question. If they had, you would know that from hooks probably. So I'm sure there's anthropologists that have the answer. Like in Polynesia, we know a lot about it from Hawaii. But anyway,
or harpooning though too, I would imagine.
Or harpooning, but. My suspicion is that bluefin, we know they came right into Cape Cod Bay, and there could absolutely be harpooning, one would imagine, because we know they've been doing that in modern, in the last 10 years. People got busted for harpooning fish that came right up to. But anyway, going back to your point about their value, what I've learned from how the bluefin fishery started was that California purse seiners, Italian families that fished out of San Pedro, L.A., north of L.A., those were tuna albacore fishermen. There were bluefin, of course, there. They were targeting mostly, the different species of tuna. I can't remember exactly why. One of them one of their seiners went through the canal and came to New Bedford or the area. Frank Siganowski, my friend, he's gone now, but he knows that history. He was one of the partners then. Leonard Ingrande, the Ingrandes were from San Pedro. They brought their boats here to fish for tunas. I believe it was also yellowfin. And it was, I forget what year, I don't know if it was 72, but the price per pound went from 10 cents when some Japanese buyer came and offered one of those companies $1 a pound, and they were shipping out of, you could get this, but once they shipped direct out of Logan, the first, and I met somebody recently that was associated with the very first plane load that went to Japan.
Out of Logan Airport to
out
Tokyo.
Of Logan to Tokyo. And when the price went to $1 or $2, I can't remember, that's when things changed.
The boom happened.
So there's probably some gray area in between. The history is not well known.
A lot of money must have been made there in the beginning. My goodness.
Yeah, but the effort wasn't super big, you know. And a friend of mine here, whose father fished for bluefin out of Gloucester, he's in the Guinness Book of World Records. I guess he caught 10 giants in one day. He wore his sport coat, and his wife came fishing, and it was a gentleman's
sport coat.
Sunday afternoon
right.
fishing, and Russell Smith, Russell's father, was in the book of, he's a very knowledgeable Gloucester fisherman. Actually, I think he lives in Ipswich. But there's a lot of lore here that we hope doesn't disappear.
Okay, yeah. I bet. They were calling them horse mackerel, and a lot of what I've seen now, that's family Scomberday. Am I saying that correctly?
Yeah.
So that'd be the mackerels, right? The tunas are in this family of the mackerels. And if you look at Atlantic tuna, it does look just like a scaled down, like a little tuna, right?
Yeah.
But what makes bluefin unique from the rest of the family? Or, is it just their size? And, what is it about them?
What distinguishes bluefin from even the other tempered tunas like big eye and albacore is that, you're right, it's longer lived, by the way. We now think their lifespan is, at least in the Atlantic, it's being estimated around 34, 35 years.
Wow.
And then
that's amazing.
They grow very fast, and their depth, and just what we talked about before, like the leatherback, because of their warm-bodied, their design, their anatomical design allows them to retain heat, and to once their muscles create heat, they have lots of red muscle. They have a very small, you would get this coelomic cavity, basically it's all muscle. So their insides, if you look at them relative to a lot of other fish, there's not a big body cavity. And so their stomach, when you finally harvest and dress a bluefin tuna, the first thing you'll say, it's not very big in there.
Right.
And their the unique aspect of bluefin anatomy is that they have what we call a countercurrent reet. It's called the reet mirabile, the miracle reet. It's a countercurrent heat exchanger of blood vessels that warms the stomach. So they soup up their digestive process. So they're an eating machine, and they can handle the rapid turnover of the food that they eat more so than any other living tuna.
Wow.
And their enzymes in their stomach have the highest sort of chemical rate of production. So they absolutely soup up their ability to eat and digest their food.
Convert that into body
energy,
mass.
into energy. And, that, and then finally, their migration patterns are such that they seem to be built for feeding on the fatty foods at high latitude, like whales or like marine mammals, whereas the say more tropical tunas, they spend more of their time in warm water. They eat mostly in the upper layers of the ocean, like yellowfin are just eating at pretty much the first, 50 meters. Big eye are also deep diving. They have some things in common with bluefin, but bluefin is just so souped up with their physiology and their anatomy and then finally, they're crossing the Atlantic as young as the first year of life. And
how long does that take?
We just know that 1-year-olds put with the tag on have been caught here the next tuna season with their tags on. And we now know that, the degree of mixing east and west or north and, basically above the equator is persistent throughout their entire lifespan.
Yeah.
And this idea that they each stay on each side of the ocean has been debunked with the technologies that we've been using since 1996.
For people listening to understand what you're talking about, Atlantic bluefin, correct me if I say something that's not right here. As I understand it, bluefin in the Atlantic were considered to be two separate stocks, an eastern and a western. And the idea was that the eastern stock was always spawning down in the Gulf of Mexico.
In the Mediterranean.
Sorry, in the Mediterranean.
Right.
And the western stock was in the Gulf of Mexico, and that there was very minimal mixing.
Correct.
And you're saying that that's sort of now come apart, and we're seeing that that's not entirely true.
That's correct. And the other part of that they were separate stocks with very little exchange, was that the Mediterranean fish were known to spawn for centuries between age 3 and 5. So much smaller, much younger fish spawning, and somehow there was this idea that that Western bluefin had delayed maturity and estimated between 8 and 12. And so,
which drove a lot of the policy.
Absolutely. Okay. And that there was separate and exclusive spawning grounds, and that bluefin always returned to the same place to spawn. So that's called natal homing. That is still the basis for assessment and management, even today, as even sort of against the accumulation, the scientific evidence that that's outdated. And it kind of flew in the face of any good scientist for a long time. How could you, once we established, I'll go back. We mentioned the Gloucester scientists that did the work in the 50s and 60s. They had it right. They disputed, the data that they put on the table was basically a bluefin is a bluefin. But the body of, say, peer-reviewed science just hadn't been built yet. They were basically naturalists aboard fishing vessels that saw what they saw, like a fisherman would, observed and did the work. And they said that, that they had an idea that bluefin probably were doing different things than had been basically the conclusions that were used as the basis for the biological assumptions for management since '81. But so that's been the framework, and the framework of bluefin is 2 separate stocks that don't mix much. That's why whatever happened in the Med and the East didn't matter in theory in the West, because there wasn't supposed to be a lot of exchange. Once over the last 25 years, my lab and our collaborators and my students were involved in a lot of the basic ecological work, and the work was also being done by colleagues in the Med and the Eastern Atlantic that showed that not only were bluefin crossing the Atlantic, they were eating kind of the same kinds of things. We established together that they grow at the same rates. And as we are now, we know that a bluefin is a bluefin. They were not 2 magically different species that had these totally different lifestyles.
Are they considered subspecies? Is there an infraspecific epithet? This is Tunis or Thunus. How do you say their genus?
Tunis tinus. I'm probably not. That's Latin, and it means rush. It means it's a beautiful name, right?
To rush. In other words, that they're fast. So they're the same species on both sides, but the idea was that for some reason on this side they matured several years, they reached sexual maturity many years faster than on the other side.
Faster in the Med and delayed in the West.
Now, real quick, in the Med, the history of fishing there, I've seen, I don't remember what it's called, this when they harvest the tuna coming in.
Matanza.
The Matanza. And that's through the Straits of Gibraltar. Mm-hmm. And they're setting up like a kind of net maze almost that they guide these tuna in. And it's quite a bloody thing to see, right? One of the things, I've only harvested a few species of tuna, but they bleed. Wow, do they bleed? And so you see the Matanza, and it's kind of shocking visually to watch it. But this must have been going on for a while.
It goes on before Christ, BC.
Okay, so in other words, even though the fishery is more relatively new, or sort of in the earlier stage of development in the Northeast, it's quite old and ancient in the Mediterranean.
Absolutely.
So they were working with the fish there a lot longer.
Not only was it the food there, but it was the basis for culture. If you read there's incredible Italian work done, and the coins that they used, the gold coins have tuna on them. The tuna was used as the brokering. Okay. The tuna, the traps, the head of the trap was like the local king, not king, whatever the right word would be, but the culture of tuna there was pervasive throughout society.
So it's like totemic to the culture there.
Absolutely.
Okay,
and
that's
trade,
really interesting.
And trade throughout the Mediterranean. It's unbelievable. It's rich.
Okay, so that's really interesting to me. So we'll get back to why this idea of the 2 stocks matters, in particular to the way policy is made. But I just want to kind of go back a little bit to the bluefins themselves. How big do they get? And one question I have in particular is you hear this term giants. And I'm curious if that's loosely or tightly defined, what that means exactly.
Yeah, that's a good question. But size is relative, right? And size matters as far as regulation. A giant here, it's sort of a U.S. term, and it refers to that the minimum size that can be harvested by commercial fishermen is 73 inches curved fork length.
Okay.
And that's the term used. They call that giant. So 73 inches and larger.
And that's, you said what fork length?
73 inches curved.
Curved.
So that just means you measure over the curve of the fish and not straight along this sort of the straight.
Oh, okay.
I mean, the rounder the fish, there would be a difference in that length.
And you do count. So in other words, you lay the tape measure not alongside the fish, but on the fish?
Yes.
So that
instead of a straight rule.
If it's a big, wide, round fish, you're going to get...
It's over so length, so not girth, but...
Yeah, but you'll get 73 inches out of a fatter fish easier than you will out of a fish that was skinnier. That's what you mean, right? Just want to make sure I understand. Okay, how big do they get?
Oh, boy, I'm going to think kilograms.
That's okay.
12 feet. I mean, there's pictures
Wow.
of fish. I have pictures of a fish landed off Ireland. The biggest ones I've seen were 1,000. We had granders that we tagged and released.
1,000 pounds.
1,000 pounds. 1,500. I'm trying to remember. There's a record. You'd have to Google it. I always forget. 1,500 pounds, I think, is a Canadian record. I should know this.
It's just
There's
when you
book.
put it in perspective, that's like a polar bear. You know what I mean? It's a very large animal.
That's
by any measurement of an animal. It's
How old would a bluefin that size be?
The challenge is that once they get to be, say, over 10 years, we can age them with, the lines, and they're like tree rings, in their otoliths. But after 10 or so, there's coalescing of the indicators, and it becomes much harder. So actually, in assessment now, the sort of the lumped category is 10 plus.
Okay.
And so old school
sense.
And growth length, they slow down in growing in length. So you can imagine the fish that are 15 foot long. You would imagine if so-called lifespan is 35 years, they're pretty darn close. And what's interesting, it's easy. The first 4 years of life, fishermen taught me this, they double in mass. So a 40-pound fish would be 80 pounds the next year. So for the first 4 years, it's easy.
Rapid
just
growth
double
at
it.
first.
Yeah, and then
down.
they slow down. And from our tagging, what we found was basically about 8 inches a year.
Okay.
Between, from when we measured, or someone measured, and then when the fish was recaptured. For the, say, mid-sized fish.
Yeah.
But remember, they start off as a millimeter egg.
As a larva.
And still the amazing thing about bluefin is the young of the year, which would be the little ones, say in the first year of life, they're rarely encountered anywhere in the Atlantic. And so
by
humans,
fishing.
So we presume Frank Mather, who's famous old, he wrote, as some people said, he wrote the book on bluefin. He had a beautiful long-term perspective on. He thought about bluefin, but one of his quotes is that the young of the year in bluefin are the cryptic biomass, the hidden biomass, and that for there to be so many, say, 2-3-year-olds, the numbers of first year of life must be massive, but we don't know where they are.
Okay, but we do know, we have a sense of how many young are produced by a single female, and the number is outrageous, right? I've read, and I can correct this if it's inaccurate, but I've read if out of one spawning, one of her offspring might reach full maturity. Would you agree with that, or does that seem excessive?
I mean, you probably, I don't know. I mean, that's the sea turtle thing that one in. Well, we're going to know sooner or later. You know why? Because of nearest kin. We're not going to know all the answers, but just like everything, just like Ancestry.com.
It's genetics.
Genetics is, the Japanese right now, they type, Japanese have 3 or 4, I think they're still on 3 generations. They can track how many survive from mothers and fathers. And so that's why, at least in captivity, you'll get information on how many. So, honestly, I don't know the answer to that.
Okay, but your point being that the genetic evidence is going to overturn a lot of observational or partial evidence that we have.
I don't know about overturn. I think it's just going to inform us about,
okay.
Like say one school. We know already that bluefin don't stick together in one school. We already know that from, not just from the air, from being in airplanes for 3 years and just watching school behavior. They're intermixing, but from the tagging, from, say, putting satellite tags, or we know that, unlike yellowfin, by the way, seem to stick together, at least in some areas, for the first year or two. It looks like there's a lot of relatedness in the school, and that a lot of that work was done in the Pacific, but bluefin don't.
Okay.
And we know that. I don't know if there's still some that still think that a bluefin school sticks together like other schools. That's not what's going on.
So two schools might encounter each other, and then they'll have an intermixing of individuals.
Absolutely exchange. And spotter pilots know that. They see that every day. For
people listening who don't understand this thing about the spotter pilots, can you talk about that a little bit? Because that comes from the fisheries industry, right? And the harpooners, is it generally, who work with spotter planes?
That's right. Spotter pilots were pretty common in the Pacific for herring and mackerel, and so it was basically another fishing tool, just to optimize your time to get to the fish and find what you're looking for. And so the first spotter pilots in New England were flying for swordfish. These are brave guys, man. They basically strap fuel tanks to their little single-engine aircraft, and they still do this. And they would fly out offshore looking for swordfish on, east of George's. And so it was these swordfish spotters that, once tuna started developing, of course they could see bluefin while they're out there. And they're also looking to get away from other fishermen. Yeah. So there are a number of reasons why, and particularly for regulations that specified minimum sizes.
So you could have a plane calling back to you saying, all right, I see giants over here.
That's
and
right.
Guide your boat.
And they're this size. And so even the purse seiners, there are regulations that purse seiners had to target fish 81 inches and larger, and there were penalties if they were wrong. And so the tops of seiners usually had a red 81-inch tuna painted on it so that the spotter would have ground truth.
Yeah.
And so, it's, it helped them be efficient. And also the seiners, particularly as the market developed, they didn't want to land all the fish at once, because then they were competing with their own fish. And so as they got more savvy
at market for price, they didn't want to flood the market with fish and drive the price down.
So they got a lot more savvy with saying, we don't want a big school, we want a small school, and we would want this size of fish because that's what the market wants right now.
Okay.
And that would be true for anybody using a plane. Yeah. And even rod and reel guys would use the plane to find fish of good quality in a different location that their competitors didn't see.
Okay.
So if you could afford the plane, some fishermen would use it for a number of reasons.
Gotta drive the cost up so much. I mean, how much does it cost to send this guy out with all that fuel?
They usually got 25 to 30% of, they were, they're considered part of the crew.
Okay, understood.
And so that's what it costs you.
They come with their own plane.
In some cases, the owner of the boat owned the plane.
Oh, okay.
It all changed. And by the way, a lot of those guys are out. They're, the guys that we worked with when we did the aerial surveys, a lot of them are older than I am. So some of them are gone. That knowledge for 30 years, we're losing that perspective from the air.
The fishing style has changed.
And the guys
this chunk baiting kind of style we see on like wicked tuna, that type of stuff.
And not just that. I mean, you can get it. I mean, like our hunters, the guys in the plane saw things that many fishermen will never see. And I have to say that I think my advantage as a neophyte tuna scientist was that I started in an airplane. And that just blew my mind that, first of all, we knew right away that the assumptions about bluefin were wrong, just from the numbers that we saw.
Okay, what year are you talking about right there?
93.
Okay, at that time, what was the assumption about bluefins?
Well, you're going back to your question, and we're easily diverted with bluefin.
I want to go down every one of these rabbit holes.
But the whole reason that the Edgerton lab at the aquarium, the industry approached Scott Krause, the head of the Right Whale Research Group, and said, we see... the industry the New England commercial fishery was facing a 50% quota reduction at that time.
By whom?
By NIMS, with the
National
Marine Fisheries Service. So the people in charge of our fisheries, National Marine
NIMS,
Federal Fisheries Regulator. They are that's the agency that sets quotas for everybody, right? Okay. Except for state.
They can only set out to 200 miles? That's our exclusive economic zone. Do they regulate outside of that?
Well, outside of that, remember, coming back to tunas, they're managed internationally under what we call ICAT, RFMO, Regional Fisheries Management Organization, which is ICAT. So bluefin and the tunas in the Atlantic are managed by ICCAT. So that's an
international body regulating highly migratory fish.
Exactly. And so each member nation of ICCAT, the U.S. is one, Canada, Japan, I can't remember if there's now more than 87 members. They basically come to the table and they both produce the scientific information that they're going to use for the assessment, and then they have what we call the plenary meetings where they allocate quota to member nations.
Okay.
And so the U.S. is a quota, Canada is a quota. So within that, then each country can decide how to manage its own quota.
Understood. So ICCAT says the U.S., this is your quota. Then the U.S. says, okay, we're going to carve this quota up for these different fishermen, fisheries, states, however that works.
Minimum size, period, season, all that.
Okay.
So at the time, the assessment for how many adult, spawning bluefin there were.
Can we say giants?
Giants in the Western Atlantic, the so-called Western Atlantic, was assessed to be very, very low, in the tens of thousands or less.
Oh, wow. Okay.
And that was based on fishery-dependent indices of abundance. In other words, the quotas the assessments were based on catch patterns.
Yep.
Of how quickly you caught the fish, which is the standard way you conduct stock assessments.
We if I just interject here, Grant and I have told the story quite a few times, but it's always so relevant. We sat down with a migratory bird specialist, an upland game bird specialist for Maine, and he was talking about assessments of turkeys. And we said, how do you determine that? And he said, we say that we take the number of birds harvested in the spring and multiply it by 10, and that's our population. And we were just floored by that. I thought, my God, that's all that you're doing? That just seems ridiculous to me that would be how you would arrive. But this sounds kind of similar. So they're maybe a little more complex, but they're taking catch rates and using some algorithm to come out with a estimated population at the time?
Well, not to get into the weeds, but two things. For Atlantic bluefin, what they call indices of abundance, they're not just using one, say, catch rate. They're looking at different areas. They're looking at patterns and trends. But it does all come back, except in one case, to catch trends under highly regulated. So in some species, if they're local, if you're all in one area, that might work really well. The problem is they're tunas.
Highly migratory.
And the problem is that you only can fish for them at certain times and places. And if they're not there, you don't catch any, that means they don't exist.
In other words, if you have, for some reason, they're somewhere else and you're not catching them, you get false readings that make it appear that you have a much lower population because your population assessment is based on those catch rates.
Well, we wouldn't call it false. We would call it, you solve that by using a long-term time series, so that over the years you're going to be right some of the time in it all.
Okay.
But there's a lot of uncertainty in any way to assess a population, and the goal is,
especially in the water, it's got to be so difficult.
Oh yeah, and coming back to what was going on, so it looked like the Western population, so-called Western, I keep saying so-called because I'm not even sure there is one, but
our population here on the eastern
Atlantic,
seaboard.
let's just call it the Atlantic, that the fish present in the Western were highly depleted. The challenge was that the industry said, we see more fish in the Gulf of Maine from the air in one day than the assessment is representing as being the entire
and the whole ocean there.
Giant bluefin. But no one would believe them.
How big do those schools get that they would see from the air?
Well, that's the fun part. So we didn't know. They asked the
fishermen knew. The scientists were not doing this, right?
Well, the federal managers whose job it was to conduct the biology were not flying aerial surveys, and at least according to the fishermen, they said that nor were they willing to fly with them to see with their own eyes.
Now you got this classic thing that goes on between regulators and the fisheries over the years. This sounds like a similar story I've heard. And sometimes it feels like, sometimes I don't understand what the regulators are thinking, and I really think the fishermen are right. Sometimes I don't understand what the fishermen are saying, and I think the regulators are really right. So I've seen this look a couple different ways.
Oh, yeah,
but in this case, you're saying that the fisheries folks were saying, hey, why don't you come along and see what we have?
Yep.
Let us show you. Mm-hmm. And they had sort of, no, this is outside of our agenda here.
If I hadn't witnessed it with my own eyes, coming in as a complete, had no background in it.
Or didn't know the politics of it.
Didn't know anything other than let's go see. And within a matter of we set up, the aerial survey equipment, the cameras. We had 13 sets of equipment, said photograph. We had restrictions on, don't take the pictures of the same school. We had ways to deal with. Anyway, we very quickly saw that they were absolutely right. We counted. I spent years in a little closet projecting the slides onto the wall. This is before digital. Jen Goldstein and I, which was my first grad student, we counted fish by projecting, and we found very quickly that schools of adult fish, we also flew in the planes just to see what was out there for those years. We had upwards of 5,000 surface individuals.
Wow.
And what we counted, what's called these minimum surface counts. Now, it wasn't a transect survey. It wasn't statistically designed. It was a see truth. So we knew exactly where the schools were photographed because we had the Loran and the GPS, etc. So it was a way to learn a lot about the real-time distribution of tuna and the types of behavior in the schools. But we counted just in the surface because that's all we could count. We had no way of knowing what was beneath the surface other than what.
How deep do you count? How deep of
we
fish
could only
see
the first couple layers,
yeah.
And that's all we could count. So what we called it was a minimum surface count.
Okay,
and
but these schools could be three-dimensional, in other words.
Most of them were.
Okay.
So it's a minimum surface count.
So you see 5,000, you have no idea if that's 15,000 or 7,000.
Yeah, I mean, the 5,000 member schools were. So you have a distribution of different school sizes, and most were between 150 and 250 fish at the surface, and but we would estimate that between 150 and 250, say 150 to 200,000 surface fish were counted during our survey times just at the surface,
Wow.
And that was just basically, this is what we counted. That was here. And that's a minimum surface count, and that's all we could do. But that information was used by fishermen attending a meeting in Washington. We weren't allowed to present anything because there was actually objections that we even did the work by the conservation members that were sitting on this committee. Nonetheless, the two fishermen that led the aerial survey or worked with us presented. They combined their 3 minutes each. They gave a 6-minute presentation to the assembled people at this meeting in Silver Spring, Maryland. And the congressional staffers were sitting in the room, and they quickly saw that there was a real mismatch between the assessment and what was actually there as presented by the slide, with the evidence in the slides. And the rest is history. That's where our Bluefin research program started. It brought all the questions to the table about what we didn't know about bluefin, which was a lot. And it showed me that, holy crap, this is a wide open area of really interesting science. And I thought, oh, I'm in.
You got drawn in there. That's where you
absolutely.
got really
and I was actually outraged by the lack of interest in getting the science right. I thought, that's just wrong.
Yeah.
And the opposition to even, we were accused, I have letters that were written to the New England Aquarium accusing us of, we're in the pockets of commercial fishermen, which you've heard that, and you know where that started from some of that. But there was objections.
Yeah, just so you know, she's pointing to a document that I have here, just sort of some of the challenges that you've had with well, you described to me this article here. This is the article you wrote.
Well, at the time,
it's called "Environmental Bullies: How Conservation Ideologues Attack Scientists Who Don't Agree with Them." It's a really fascinating read.
Well, if going back to the history of, the whole idea of cooperative research partnerships with fishermen is what the old guys did, and there was nothing wrong with that. But somehow in the polarization of sort of environmentalism as far as bluefin tuna, there were many problems with bluefin tuna. There was overfishing, there were pirate fishing. There's a lot of bad stuff going on in the Atlantic.
Still or then?
Then, in the 90s. And there was...
What countries were doing the pirate fishing?
Well, I don't want them to come after me right now. But
what regions of the world?
The Farallon, sorry, not the Farallon. Not the Orkneys. Off the coast of, they're big fishermen, they're whalers off the coast of Scotland. Not the Orkneys. Starts with an F.
The Faroe Islands?
Faroe Islands, thank you. In the early 90s, I forgot what year, but the Faroe Island, they had no quota or allocation. They landed more bluefin tuna, pirate-wise, than the combined Western quota of Canada, Japan, and the U.S.
No kidding.
And then there was overfishing throughout the Mediterranean.
They would sell into the market.
I don't know much about the market,
Right,
but
okay.
and then of course Gaddafi and his brother. There was all kind of really malfeasance going on in the Med, Africa, everywhere. It was a mess. So the conservation policy people, they focused mostly on the U.S. They I guess had a feeling that they wanted to protect Atlantic bluefin. That's admirable, but I thought that they were focused in the wrong place. And the science that they were sort of backing is, what was the reality, we just showed very quickly was wrong. And we also became the enemy, I guess, because I guess the agenda was to get to what you wanted, which was an endangered species listing for Atlantic bluefin.
Okay, I want to break that apart a little bit. So you're talking about you've got these environmentalist groups it's interesting being a hunter, because I find that there's this idea of a conservationist and there's this idea of an environmentalist, and they sound to the untrained ear sort of similar, but they kind of break apart into two parties, and they tend to be, if I was going to characterize it, I obviously am hardly an authority on this, but I would say environmentalist tends to take the hands-off approach, and the conservationist tends to say we can have a sustainable harvest of a natural resource. That's what I tend to see. It sounds like it's something like that here, where were they just wanting to shut it down then?
There's so much complexity in all of this, but I will say this. I mean, going back to what you said, I was trained in the early 70s. The academic environmental movement started, where training biologists, geologists, and others to think about, the challenges to the earth and sustainability. And I was lucky enough to be at the University of Pennsylvania, that has a famous urban. It was Ian McHarg was the urban development, and then also the one of the first environmental studies departments that basically you had a double major. And what I learned there, my mentors as an undergrad taught me to come to the table, just as you said, and come to solutions to allow for use, because we're humans, we're going to use it. So honestly, that's deep in my training as an academician.
Right.
And so it just, and I'm a sea turtle biologist and conservationist. Just to say, I did work in sea turtle conservation. They're truly endangered at the time. Right. So it was bizarre for me to sit in, to get these letters from so-called environmental or conservation sort of representatives that were challenging the fact that we were actually doing scientific work to get better information. That blew my mind. And then to be even attacked personally for doing science, letters that said some right whale volunteer was misrepresenting, and the aquarium was called out they were members of a conservation group. They were chastised for doing the work. I mean,
for doing science.
I mean, that to me illustrated, this is the new conservation, not conservation, it was advocacy. That's different.
In other words, the ideology trumps the science in their minds. Like, we don't want the real science because the agenda is more important.
The agenda is important, and this, I don't know. I just, the whole thing of the honest advocate, it's tough. I mean, I have my own biases, of course, and but as scientists, we're taught the scientific method, and so it was a real shock for me to learn
that they didn't want your data.
Didn't want the work done.
That's
and if
crazy.
you worked with fishermen, you were already tainted.
Right.
That is, to me, I mean, if we don't work with fishermen, or farmers or anybody else, you have to be nuts to think that you should not be working together. So to me, I'm an outspoken sort of critic now of misplaced advocacy, because there is scientific consensus if you look closely enough.
Okay.
And in bluefin, it's there. It's not necessarily represented right now in current management, and it's hard. There's a big lag time, and also it's all about now the less money. My lab is out of funding, so all of the stuff I
learned about your work from an article by Billy Baker talking about you doing lab work out of your garage. Could you kind of put that in perspective for us, so that, and you work. Tell us where you work now, and sort of what's happened with your funding over the years, because I think it's a really important part of the story. Well,
what I like to highlight first, I mean, I'm known for getting to the bad stuff first, but I would point out that
you haven't seemed like that so far.
My lab, I'm really proud of the work that we did together with my fisherman partners, they basically pointed us in the right area to go after the scientific questions that I never would have known about without them saying, hey, you better look at this. Then the congressional support we had when there were federal earmarks to build the University of New Hampshire, the Large Pelagics Research Center, that supported all of the PhDs and the master's and undergrad students and postdocs that we were able to train and get them into the field, or not, if they got out of it. And then the chance to work with international colleagues, because it was tuna, it was international.
Right.
So I've been going to Spain, Madrid, and working with the Basques. I mean, one of my greatest thrills is that we were able to work with some of the best fishermen in the world in the Atlantic, the Basques.
And your work's taken you some cool places, huh?
Really cool places. We did it because we had the money.
Yeah.
My students that got trained was because we had the funding. And unfortunately, you know the reality of life. Things change, and universities have to have the money for us to stay there and work there. I was never tenure track. We always had enough sufficient grants and contracts and federal support for the fisheries science, and particularly from the industry that said, hey, we need this work. We were funded and we competed well on money that was put into the NOAA or the federal budget so that,
NOAA, just for listeners, NOAA is the National Oceanic and Atmospheric Administration, right?
Yeah, and so we had a good run, but the challenge was that once the federal earmarks went away that supported my lab at UNH, we moved back to Gloucester to try to, we'll redo this. We'll try to rebuild in Massachusetts, where there's Massachusetts is a bluefin fishery.
Hey, we'll get right back to the interview in a moment, but first, domesticated, civilized living restricts the natural movement of your human body, forcing you daily into chairs, cars, and rigidly defined pathways. But when we head out onto wild landscapes, we carry these dysfunctional, domesticated movement patterns with us. MoveNat is a natural movement system that trains you and your body to move like a human animal again. Are you interested in building natural movement skills that'll improve your physical capability in the field and in everyday life? MoveNat's new Level 1 Fundamentals e-course is structured to help you re-educate your body using practical, natural movements that enhance fitness, function, and physical capabilities, all from the convenience of your home. Learn to move the way nature intended. I attended MoveNat's Level 1 and 2 trainer certification courses a few years back and came away with practical knowledge and skills that I use every day and that have changed the way I train and move across the landscape, vastly improving my overall mobility, coordination, and fitness. This new e-course is movement-rich with clear instruction. Its progression-focused methodology takes you from restorative mobility exercises to challenging movements. All ability levels are welcomed and accounted for. Enroll now at wild-fed.com/MoveNat for instant access to the full e-course. Again, that's wild-fed.com/MoveNat, MoveNat. Again, wild-fed.com/MoveNat for instant access to the full e-course containing 16 classes designed to be followed over 4 weeks with specific and measurable movement goals for each week. The course is self-paced and you'll receive lifetime access. MoveNat. I can personally recommend it. It's a fitness and movement system that's designed for the wild. Now, back to the show.
I lived in Gloucester. We wanted Gloucester to continue to thrive as a fishery science center. So that's when, and that's not even worth going into, but basically Massachusetts is a pretty political place. And as far as where you work and timing is important. And so our first 3 years trying to reestablish the old the UMass Marine Station, that was an important place in the past for fisheries and for fish. That was Herb Holton's lab. We made a really strong effort to make that work and to bring our ICAT colleagues here. And we had the Basque, the French, the Israelis, the Spanish. We had everybody here, but the politics didn't line up and the funding didn't line up in the state of Massachusetts. So we lost our backers. And
is it partisan? I'm trying to understand what you're saying.
No, it's just there wasn't enough. Massachusetts wasn't senior delegation. So in other words, there wasn't enough federal money around to sprinkle what they used to call pork. Pork actually paid for good science throughout science, medical research.
Pork is this money,
wasn't all bridge to nowhere.
That you get at the top?
Pork paid for a lot of our science that we put on the table.
Okay, and that money's dried up.
Dried up for us. dried up from Massachusetts.
Even okay.
And then now it's going to other states like Mississippi, Alabama, Maine, places that.
With tuna being such a contentious issue, I'm surprised by that. Where it's in the public eye. I mean, I feel like I see an article every week about it, about the fishery.
Yeah, you do. But I mean, the good thing is that it's not a crisis fishery anymore. It's good. I mean, that's your story.
Interesting that you say that, because that is what I think a lot of people listening to think this is a crisis right now. And that's what's so fascinating about your work to me, is that you seem like this kind of, and forgive me if this isn't right, but you sound like this lone voice saying, hey, this isn't actually, because everything else I've seen, in fact, Grant and I have dreamed up an episode of our show where what I really want to do is harvest a bluefin, and I really want to serve that out to, 40 people, let's say,
sure,
and have this sort of, seat-to-table meal that we put on, and I've worried about how will it be perceived by people because of all of the issues around bluefin. And then I thought, well, if we're going to do it, we should do it soon before they shut this fishery down. And I've had all this in my mind. And then I started reading your stuff. I was like, oh, wait a second. Maybe none of this is actually how it's been portrayed.
Oh, no. And, I mean, we're the lone roof on some topics, but if bluefin were not now getting very closely rebuilt, and there are reasons why I think they're still vastly underestimating what's out there for all the 25 years of work, because I think there's a hidden, like the cryptic biomass, there's a lot more bluefin than anyone's counting, the dark matter bluefin. But if it weren't, you could not have a commercial fishery in the U.S. The beautiful thing about U.S. fisheries, as many NOAA scientists have talked, they've rebuilt through regulation and cooperation, the vast majority of our U.S. fisheries. Bluefin tuna is getting up there, and the real challenge is not our quota allocation, the fights in between. And the quotas are still held to what they were, more or less, going back to 1994. So there hasn't been any increase for the most part, other than
in the take.
In the quotas, in the takes. So they're still fishing under highly regulated management.
Do you think that should be loosened, or do you think that...
I think first the science should get to what's better.
Let's talk about that. And going back a minute ago, it sounds like some of these environmental and conservation groups thought they had like a sort of buttoned up case, right? Like they were just gonna push this thing towards. Now, would it be ESA or is it CITES? Can you break
all those?
Okay, so ESA is the federal. That's our Endangered Species Act. CITES is international, correct?
Yeah.
And what does that stand for? It's the
the Convention for the International Trade on
We harvested
an
an alligator down in South Carolina, and we had to have a CITES tag for that. And I think because there's international trade in the hides and things like that. Mm-hmm. So that was where I first encountered the term. So that's a member nation thing. That's like an international version of this endangered species thing, right? So also the IUCN has them listed as endangered and on their red list, correct?
Correct.
So if I go to like the Wikipedia page on bluefin, I'm going to see a little thing like endangered, because they always list.
That's the problem. They don't distinguish between Atlantic bluefin and Pacific bluefin, and that's sometimes I think it's on purpose. They're different species. I'm not species. They're totally different assemblages with totally different populations. Atlantic bluefin tuna are nowhere. Atlantic bluefin are pretty close to being rebuilt.
Yeah.
And they're not a stressed population as the Pacific. Where I think the Pacific is where we started in 93. There is a huge amount of science that hasn't been done yet. So there's a lot to catch up on out there. So the representation of the Atlantic as being the same as Pacific and the generic Atlantic bluefin are overfished and chefs shouldn't serve them and all that, that's basically obscuring the reality that they're not at all in the same boat.
Okay.
And I think a federal fisheries manager will be able to tell you that. And so I call into question, for example, if you read an AP story or something, when they lump tunas and they don't get it right, that's why the public doesn't understand or has fears. And of course you'd have fears if you thought they were endangered. But I mean, that's the challenge, as you know, the complexity of the story. The news or the media wants to break it down into something simple, consumable sound bites. And I know, I get it. But in the case of these complex issues, I mean, Atlantic bluefin tuna is being fished sustainably by the U.S. and Canada and Japan. Period.
And Japan as well.
Yeah, I mean, they're all under ICAT as far as, and so that's the story. We wouldn't have a fishery if that weren't true. It would be shut down.
Okay.
That's it.
Okay, so they were pushing for this sort of endangered listing, and then you
Repeatedly.
and your folks started to bring in kind of you got aerial photographs, so it's kind of hard to disregard that, and then that kind of throws a wrench into this kind of they thought they were fast tracking this thing to shutting down the fishery, essentially, right? Because that would have shut down the entire fishery.
There are still goals to reduce.
And that's positioned you sort of, that puts the target on you a little bit and on your lab or team and the work you guys are doing because of...
Well, we didn't, honestly, we didn't pay attention to that.
Okay.
I mean, now as, all these years later, when I see it's still going on, even though we put all the peer-reviewed science on the table, by the way, it's almost like political stuff. It's just ignored. I mean, you could have like even the age of maturity, which they're still calling debatable. Well, that's a joke. I mean, how many times do we have to hit them overhead? Here's peer-reviewed science by experts. And you're saying your opinion is equal about the age of maturity. Oh no, you still think that somehow there's magic delayed, maturity, or that. Anyway, it's just, to me, it's just stunning. Science didn't used to work this way.
Well, it still technically still doesn't, or it's not supposed to, or it's not science. This idea of the delayed maturity gets into this idea of another spawning area, correct?
Yeah.
I want this to come across as simply as I can because I know this is like you said, highly
That's why
nuanced
Billy
stuff.
Baker got out of this whole story, by the way.
Yeah, it's too complicated, right?
I mean, if it's about food, we could talk about the food side of it, but I see that you come at it the whole way. But yeah, it's
Well, luckily we have this long-form opportunity, so I think we can sort of break it down. But the idea is that now we can tag fish. GPS satellite, right? So we can
not GPS, but
it's not GPS.
A fishery independent tag that allows you to get the information off the fish without having to kill the fish. But it's not like a shark or like a turtle tag. It's because because most of the fish, tunas or billfish, spend so little time at the surface.
The GPS wouldn't work. They'd be too deep.
Like longer than a second. They do come up, you see, from aerial surveys. But
you'd have no data once they dive if it was just GPS.
You data log, so it's recording. Okay. So it's a data logger. And so it actually, the guts of the first pop-up tags were actually a bird transmitter, what we call micro PTT. And so the bird guy, who's actually had the same PhD supervisor as I did, who Paul Howie developed, basically put his micro transmitter in a pressure-resistant tube and developed the first pop-up tag. This is not their tag, but it's basically a computer. It has a light sensors and depth pressure sensor and a temperature sensor.
I wish I could show this to people. I'll take a photograph and we'll post it. But it looks
sort
of like it almost looks like you'd plug this into your car lighter
plug
thing. Yeah.
Oh,
yeah, right. So what part of this is actually attached to the fish?
When a fisherman found it once, it's described as an egg, a cigar with a whip antenna. Yeah. But we make tethers, so there's a mono tethered. I adjust the length of the tether to the size of the fish, and then there's a monofilament dart. So they're they're positively buoyant because this is syntactic foam float rated to 2,000 meters. So they're designed to record. The sensors are there. The data, the chip, they're recording the sensed information. Depth and temperature, or pressure and temperature, are straightforward. But these are light-based geolocation, so that means that there's light sensors, believe it or not, in there. And so it's like celestial navigation. We estimate the dispersal pattern of the fish based on the estimate of sunrise and sunset, which is based on basically the light level curve. And for each place on Earth,
approximate coordinates.
Approximate for latitude and longitude.
Wow.
So there's a clock, so you know precisely what time it is.
Right.
So the longitude relative to Greenwich Mean Time, basically sunrise, sunset, civil sunrise, sunset, and then of course latitude. You have your day length, how long your day is. So it's kind of an ancient.
Yeah, but you're going this much light at this time. They must have been here.
But then they're at depth, right? So it's a science because we also use the sea surface temperature recorded precisely by the tag.
Okay.
You basically have an area that you think it could be. We use the depth, so we put constraints around the estimate based on light levels. And then you also have what's called an ensemble common filter approach. So you basically take all the data that you can use and you use assumptions based on your previous estimates of how quickly you moved from point A to B. And so it's ensemble. So actually, you didn't meet my partner, my colleague Tim Lamb, who's working in Korea right now on Patagonia toothfish. But Tim Lamb is among the developers of optimizing the data that these tags carry.
Incredible.
So they're $4,000 a piece. It's 1996 technology. The price hasn't come down, nor has the technology changed that much.
Wow. Okay.
Which is a challenge.
That doesn't look or feel in the hand like it's worth $4,000. So those tags gave us the ability to determine that not all the fish were returning they didn't have that like spawning site fidelity that we were told that they had.
Well, step one was these tags don't tell us when a fish spawns. So that's the first challenge. But you're right. What we learned, the first paper we published was in 1999. The first tags we put out on the fish out of the Gulf of Maine, when their tags came off, and that was before we had...
they just degrade that mono UV breaks the mono down?
No, they're actually programmed. I'm sorry if I had the other thing ready. There's a signal. So say the due date. We usually set them for a year. So when the clock ticks on that day, a year later,
breaks away.
It sends a charge. There's 18 gauge or there's wire in here. In the presence of seawater, it gets the charge. So it gets charge induced to it. So the nose cone pulls apart from the tether.
Cool.
And the tags floats up to the surface and starts transmitting. If the fish dies, say there's post-release mortality, which by the way there's almost never in bluefin, the tag is programmed to come off at the fail-safe at 1,300 meters or so.
That fish was sinking
If the
down.
fish were dying, yeah. So you don't lose the tag when it goes below.
So you still recover your data.
Yeah, and then the other thing is if the tag sheds, it pulls out of the fish. If it's at the same depth or at the surface for more than, say, 2 or 3 days, it's programmed to talk.
Okay, yeah.
So you get the data. So if the fish dies, you know
Do you have to recover this to get the data?
No.
You can
so
pull the data from the satellite.
This is an EPIRB, basically.
Okay.
So there's a company called CLS. It's a French company. And we basically pay to have the data distributed to us. So they put their receivers on NOAA satellites. The satellites are going around, so just like an EPIRB system.
Yeah.
So we pay to get the data transmitted to us, and then there's a whole lot of science or
that has to
programming to get the data off, and so that's how they work.
Okay.
So, but if these land up on the beach, the beautiful thing is if you do get them back, they're not designed to come back, but if you do, you get a lot more data because they only can send so much bandwidth. Right. Yeah. So you get data almost every 2 or 3 minutes. So you get much more data. That's why the reward for recovery of a tag is up there because they're still valuable.
So if I see one of these on the beach, grab it.
Definitely. We've gotten them back from Scotland, from all over the Atlantic. Twin 8-year-old girls found them on the Argyll Islands, from Labrador, Newfoundland. Where else? Wales, and just all over the place. That's in the Med, and sometimes the fish are caught with the tag still attached.
Yeah. Okay, great. That makes sense. So once you had that data, how did it become clear that I'm trying to figure out a way to like tie this together that our fish, I say our, here on the western side, were not always going down to the Gulf of Mexico,
right.
which made it appear. This is where the assumption, they must spawn later, though.
Yeah.
Can you kind of like tie that? I'm not saying it well, and I want people listening to understand what I'm trying to say.
Well, if you say the assumptions that all the, say, our so-called Western, our historic fisheries were targeting Western Atlantic fish, that their life history story was that they're all supposed to spawn in only one place, the Gulf of Mexico. Were known to spawn in only one place, as assumed by, fisheries management, and that the fish that are found there were big ones and old ones. So you could see, well, if that's the size of the fish on the only known spawning ground, then that must be where they all spawn, and they must be that big and old before they're sexually mature. And so we didn't know until we started putting these fishery independent tags on, so you don't have to catch the fish. The first years that we put tags on with fishermen, the tags were only a percentage of the fish were indicated as to having gone into the Gulf of Mexico during the known spawning period. Most of them were outside of the Gulf of Mexico. So over all these years, it became very clear that 20 to 30, maybe 20 to 35% of our each year sample of the tags that went out went to the Gulf of Mexico during the late spring, and the rest of them were in the Atlantic.
Oh, so as much as 70% of the fish
were outside.
The western fish were not going there
70%
during the spawning time.
Of our tagged
fish.
And we assumed that the fish that we were tagging represented a cross-section.
Right. Yeah.
And so we said, well, wait a minute. Because we were studying also the physiology, I mean, that's the thing. We didn't just look at the fish going to where do they go. At the same time, because of my focus on physiology, our focus was on ecology. So we're looking at what are they eating, how much fat are they putting on their body, and we also looked at how much fat they were putting on their gonads. And the fat that they put on their gonads, which no other tuna does.
Oh, they're the only species that does that?
The only tuna. I don't think there are many fish that do it. I know there's one other little fish somewhere that puts fat on its gonad. That fat, it's incredible. It can be a 10-pound fat pad. That is used to make the eggs and the sperm. Yeah. And that's basically, they take their load of their energy and they take
that they accumulate through this hyperfeeding time.
Exactly. So when fish leave here...
On herring, if they can?
Herring, mackerel, squid,
oily
salmons,
fish, yeah.
They put this incredible fat. You should see the fat pads. I mean,
I saw some photos in some stuff because I was looking at some of your work online. So I'll include some of that in our show notes if people want to see them. But it looked like big footballs of fat.
And it's, to me, it's incredible. I mean,
I want to eat that.
Nobody studied. It's just pure
fish oil.
Fish oil. But anyway, so it basically said to us, so people answered, well, they don't spawn every year. So that only these 20 to 30% are spawning. And that would make sense because it looks like the population's so low. So wait a minute. That would violate all life history rules for tunas. And secondly, hey, by the way, we did the work. We can show you that when fish leave here, they're leaving with these incredible fat stores. So there's no biological sort of...
So you're saying, hey, I know these fish have all the fat that they need to make Milton roe.
Exactly.
And I know they're not going to the Gulf of Mexico.
They have to be spawning somewhere else.
And people are like, oh, no, they're just holding it, and they're not going to do it this year.
Right.
Which is like...
or for 2 or 3 years.
As a biologist, you got to be like, come on.
What are you not? And what's interesting is we knew we were right. You know why we knew we were right? We knew we were right because, not because of what we had done alone, but the guys that I mentioned early on, Pete Wilson, Frank Mather. There's a famous quote that I showed last night. Frank Mather said in 1965 that the oldest, biggest fish spawn in the southern areas in the late spring and summer. The smaller and the medium fish spawn in the Atlantic later in the season, and is like up on east of George's Bank. And I thought, holy crap, that's a little radical. And there was never any evidence.
When was he saying that?
Frank Mather in the 60s.
Okay, so he's saying that there's a northern...
Southern big old fish spawn in the south.
Right,
first.
but then he said smaller fish were spawning in the Atlantic. Later, and then later the so-called science came in and said, no, that's debunked. We know that they go here.
No, they just ignored
it.
it. They just ignored it.
It was never taken seriously.
No, or it was suppressed.
Okay.
Some people, the old-timers said it was suppressed.
Okay.
It wasn't convenient. And the other reason why we knew that it was correct was because my Japanese colleagues that knew bluefin better than anybody in the world, my Japanese scientific colleagues would sit next to me at meetings at ICAT and just roll their eyes. They knew. They showed me that they knew the spawning areas in the Caribbean. They showed me historic maps of the Japanese. So we knew that if the Japanese experts on bluefin biology tell me that this is the way it really is, I'm going to listen to them because they're really good at bluefin science.
Okay.
And so that's where the politics came in.
Yeah, but that's inconvenient to what we were talking about before. If you're pushing for the CSA listing or the CITES listing, and you know there's another spawning ground, and you know that these fish are spawning earlier in life, that starts to impact the regulation a little bit. So it's inconvenient if that's your agenda.
I think early on it wasn't that. It was that they, in their defense, I don't think their job wasn't to be a good scientist in biology. That was not what they did. They became, they were representing policy advocates. So they were, most of them didn't have the expertise per se.
To even interpret the data anyway.
Or frankly, we hadn't done the work then. It was done earlier and it was ignored, but it's almost like it had to be done again. And it had to be done on a different scale. And so you're right, though. And so you could see that a lot of that work didn't come into peer review until Gilad Heinisch, Jessie Knapp, who did the work on the fecundity. She worked in the Gulf of Mexico fish, but 2 PhDs in my lab worked on maturity and reproduction. And Gilad Heinisch, who was an expert from the Met already when he came to my lab, who was looking at reproduction in the across the Mediterranean, his master's work showed that bluefin spawned over a much bigger area in the Med, and that they had size and time-specific spawning patterns from the west to the east. And so all this was starting. If you start looking for larvae, you find them.
That's what I want to talk about next. So it reminds me of the story of American eels, where it's like trying to figure out where they spawn, and really what you have to do is follow density of larvae until you find the densest. And then you got to know, okay, if this larva is this old, it could have only have gone as free cast this far, right? Do bluefin larvae, are they free cast? Or the eggs are free cast?
Yeah.
Okay, so if you got to find the larva, so you find the larva, and if based on the age of that larva, it can only have come so far.
That's right.
And so what happened in 2016?
Well, so we've been predicting, we lowered the age of maturity, we weren't popular for that. In fact, it's still contested, even though it's established.
And you lowered it to say, hey, these fish are sexually mature the same
around
age
the
that
same
the ones
age.
Are over
and there's individual variability.
Sure, of course.
And there's still more to do.
Like any people or animals?
Yeah, just like that. But there's more to do there because we didn't sample many 4-year-olds. But anyway, coming back to what were we talking about? The
larva.
The larvae. So we got lucky in that other really good scientists that happened to work for NOAA, National Marine Fisheries, in the Northeast Fisheries Science Center. We were lucky because the senior scientist now, the head of the Woods Hole group, John Hare, and Dave Richardson, who was the lead in that study, Richardson et al., they're larval ecologists. And actually, Dave and I went to the same grad school at the University of Miami. They're larval ecologists of tunas and billfish. John Hare was on Jessie Knapp's committee, my grad student's PhD committee, and he said to me, even though he didn't work on tunas at the time, he said, he said, I know you're right about the maturity in the larvae. He said, he's a scientist that knew his stuff. And so, that wasn't his job at NOAA. Dave Richardson, who was also a PhD in larval ecology of tunas, who works at Narragansett, so he's in the Northeast Center, he was always intrigued by the lack of anybody's interest in looking for bluefin larvae where we all thought they were. And in fact, that it was held to just the Gulf of Mexico and then finally moved out to the Florida Straits off Cuba, because we'd already been going public with, wait a minute, this is where fish are going. And so he was basically going to look for himself. And so Dave was out running. He does an awful lot of other fish work, but he was on a larval cruise looking for whale forage. So
they're pulling a very fine mesh net.
Larval toes, and he puts the appropriate toes in for scombrid larvae. And in the very first toes, when he finally picked the larvae, he said, "Oh, here's bluefin right away." And
where was he?
This was in the Slope Sea, so that was the number of stations that you see in that paper. Yeah. And in the press
just for
release.
people listening who are like geographically kind of, we're talking about
offshore.
Off the coast of the Northeast, basically.
Right, east of George's Bank.
Yeah.
And
so directly east of which states?
So that would be from, say, I would say Maryland up to the Canadian EEZ.
And so this is interesting to me too, because when I picture the Mediterranean and I picture the Gulf of Mexico, these are like inland marine water bodies. I would imagine that they are a little bit isolated from the sort of chaos of the sea. This idea that they're spawning also just sort of between the Gulf Stream, offshore,
edge of the Gulf Stream,
but inside.
Not in the Gulf Stream.
Yeah, but I'm saying inside of the Gulf, between the Gulf Stream and the coast.
Well, the first cruises were actually on the other side, on the pelagic side.
Oh, okay.
And so if you think about what we call the Gulf Stream recirculation areas, so all these eddy systems that are spun off, that's where a lot of fish spawn, including big eye and marlins and
because of temperature, but also because of movement.
Right, gradient, and there's retention areas because of these basically loop eddy systems that were well known for other tunas, even though the other tuna larval areas are very poorly known, except in off Africa. So there just hasn't been a lot of high seas exploration for larvae or for early habitats. But anyway, Dave Richardson and his colleagues, collaborators that were on the boat, they found the scombroids, and they had very limited opportunity to look. So it's not like he was able to design surveys as they were being done in the Gulf of Mexico for decades. So this was what we call opportunistic sampling. Yeah. So even with limited sampling, Dave Richardson and colleagues were able to find the larvae, measured to DNA. They had blind DNA testing. The samples were sent up to Alaska so that the people doing the genetics analysis didn't know what species. They correctly identified yellowfin versus bluefin and other species. And so all that work was done because they knew that it would be so controversial and so challenging that sort of an outsider, even within the NOAA group, would decide to focus on bluefin. They weren't supposed to look at bluefin. Yeah. And so we learned from them because, particularly because the senior scientist on the study had been on my students' committee. We got together and he said, "We've found what you've been looking for." Wow. And not only had they found it, they had done a lot of really great work on the larvae themselves. And you could see that when we published it, our paper was reviewed. We know 2 of the reviewers out of the 3. They're very highly placed, famous fishery scientists that knew that the paper was controversial. Yeah. And the paper had been rejected by Nature and Science as not of much general interest, even though the papers that found other things were published in Nature and Science and covered around the world. Of course. So, but they're
on board with that basic agenda.
They said,
would appear to be from the outside.
They said that it was in the Proceedings of the National Academy of Sciences, the PNAS, a very well thought of journal, and there were letters of protest that were submitted. They're not peer reviewed, the letters, but so the controversy. They said, "We don't agree with your findings. It could have been a one-off." Of course, one of the NGO leaders that Carl Safina represented, that they must have been basically floated out of the Gulf of Mexico, and we were wrong about what their origin was, and there were only a couple larvae found. And over the next 3 years, we basically dissembled all of those complaints. But then, of course, there was never any more funding to go out and do any more work.
Okay. So where does that leave the state of the science on? So again, to wrap a bow on this for the people listening, there was this idea that bluefin in the Atlantic had 2 spawning grounds. Now we know that it looks very much like they have a 3rd spawning ground near the Gulf Stream off the eastern seaboard of the United States.
Honestly, we still have a lot to do about it, but I would phrase it. That paper was sort of conservative about what it said about bluefin spawning in the Atlantic. It was only where they sampled, and that where they sampled was the Slope C. My personal opinion, it's an opinion based on what the satellite tag data and what I know, and then we look at the habitat to establish. We've even done work with our French colleagues, Patrick Lahode, who's one of the top known larval habitat modelers. We basically said, okay, this is bluefin larval habitat. Where is it? At what time? And Patrick has been modeling tuna in the Pacific and the Indian Ocean and the Atlantic. So basically, before we had larvae, we predicted where it should be and when, based on larval habitat, like you said, the Med in the Gulf of Mexico. The Gulf of Mexico is basically, the loop current is an extension. It's a Gulf Stream. Yep. So you're right that they're enclosed areas, but all the other tunas are spawning in the Atlantic. Billfish are spawning in the Atlantic. Okay. So what everybody thought bluefin are special, what I think we're finding out is bluefin are not that special when it comes to
where they spawn
as far as their needs. What blew my mind was that they would spawn so far north, high latitude. But what we now know is, hey, it's high latitude in the summer. That means longer days, and east of the Gulf Stream, warm water. So you essentially have equivalent habitat as the Med and as the Gulf of Mexico at their times of spawning. Yeah. So I think that it's a continuous potential habitat from the Gulf of Mexico or from the Caribbean, which is where the old timers found them spawning, all the way up to the Canadian EECs.
As long as they stay tucked in along that Gulf Stream, they have the warm water.
As long as they're in the right habitat.
Yeah, okay.
And it could even be in the central North Atlantic. We first said maybe the Sargasso Sea retention areas, but that work needs to get done. And that's the problem is, where are we now? We're in the dark because there's still no priority or funding or planning to do anything, to my knowledge, about getting back out there and really doing it right. And that's the problem.
Okay. And then how strong are the forces still fighting and pushing for the listing?
I don't know, because honestly, after 25 years in this, you get tired of, and particularly after you do a lot of scientific work and it doesn't seem to move the ball.
Yeah.
And it seems like the sort of opposition to getting it right seems to be, I can't understand it.
Yeah.
Why they're not advocating to get to the nature of the Atlantic population. So I'm stymied, and in that
it seems like they're more interested in ignoring new science and
or
just
saying it's
pushing,
wrong or debatable.
Okay.
It's
right.
No, well, let's see your science. Well, you don't have anything to counter it. So I'm just befuddled by it all, honestly.
But we'll continue then for some time to probably see these stories. It's like you joking about wicked tuna, but it's like the show starts off with a little disclaimer, like, bluefin populations are in peril in the Atlantic.
That's just wrong.
Yeah.
Just wrong.
For people who are listening to this who are like, well, can I still order bluefin? Can I, ethically, obviously you physically can, but can I go to the sushi? Can Grant and I leave here and stop at a sushi place and get bluefin and feel okay about that? Or
yes, absolutely. And chefs, when I work in Hawaii too, I meet a lot of chefs just because we're interested in fish, and almost every chef will say, particularly sushi chefs, I don't buy bluefin because they go for Seafood Watch, various places, and I don't understand why they're not updating some of those listings, like they don't eat bluefin. But the challenge here is, let me just answer yes, emphatically. I eat bluefin tuna, or would. I think it's healthy. I know that we've done a good job of rebuilding and protecting the Atlantic population, finally got some teeth to stop the pirates and the illegal fishing. I mean, there's problems. We do have climate change. We do have ocean warming. We have other challenges that all the species are facing. But as far as our fisheries, I think they have been fishing sustainably for quite some time and are being intensely monitored. All that contributes to, if you personally want to eat a delicious bluefin tuna, I would say go ahead. The other important thing that I think your listeners might want to know about, which is something that I have experience with, and that is the portrayal of the big fish, the older fish, swordfish tuna, some
Mercury.
Sharks, as methyl mercury as being bad for you.
I didn't want to bring you down that trail,
no,
but I'd love to hear anything you have to say about it.
Well, it's science. Curious. I mean, the good thing is, if you go in the Pacific, it's been well known for decades that populations that consume a lot of fish, raw fish, or the tuna is billfish, they don't have methyl mercury poisoning. And so there was no medical evidence in populations other than when there was mercury spills, or the Faroe Islanders, who, by the way, were eating whale meat. The reality of sort of the toxicity of methyl mercury is toxic.
Right.
As far as why we don't get sick from consuming like I have, I should be twitching and glowing as far as bluefin consumption, is that methyl mercury is bound in molar ratio by selenium.
Yeah.
Most of the marine fish, not freshwater fish, you can actually have some trouble eating big old pike because of their methyl mercury. Native American cultures in the Midwest have challenges with methyl mercury poisoning because there's no selenium in the...
Selenium basically washes out to sea, right? It's like a lot of our soils are depleted in selenium because it migrates down into the ocean. But marine animals will be rich in selenium. Yeah,
and so the explanation is that, by the way, selenium can also be toxic in high. But what the work that's been done by many labs, including the National Institute of Health, and I'm going to get to this, what they found was that the reason methyl mercury doesn't seem to pose actual toxicity is that it's bound by selenium.
By the selenium.
And so that in species that have selenium, present. So
in the digestive system?
No, in the brain, in the fatty tissue.
It happens, the buffering, the selenium buffers the methylmercury in the fish or in your body when you consume it? Okay, so it's already buffered or already bound to selenium.
It should be bound to selenium in molar ratio, and if it doesn't have selenium, then that could be considered a problem if you have high methylmercury, which would be the freshwater fish.
Right, understood.
Whales don't have selenium.
That's why the whale meat is so much higher in it.
That's why the Faroe Islanders that had levels. But so the study, so we've always been up against. There are labs, very high profile, Ivy League labs, toxicity labs, that will say that eating seafood is dangerous. The NGOs say eating tuna is dangerous because of methylmercury, even though the studies have shown that that can't be the case, because otherwise all these populations that consume large numbers and their children and their infants would be sick, and they're not. So the people that have been, there's some famous cases of people that say they eat toro every day, or they're wealthy, they go to New York City, they go to a very high-end sushi, they get sick, but they haven't been screened. It looks like the challenge has been that it's easy everybody knows that eating tuna or toro, you'll get methylmercury poisoning. They weren't screened for any other issues or heavy metals, or there could be neurological diseases. So the assumptions by many, like basic health practitioners, they're not experts in the field, and they say, the guy said, I ate so much sushi that it must be mercury poisoning. They look, they find high levels of mercury, and they say, you've got come back to the studies. They're demographically over large scale studies, and very recently, just a month ago, there's 16 authors, or many authors, on a paper led by Commander Joe Hilbaum, Dr. Hilbaum. He's the head of the toxicity group at the NIH, and many other collaborators finally had the biggest review of the impacts of methylmercury on
do you know what the review is called?
I'll send you the link.
Oh yeah, please. I'd love to post that with this interview.
But we've been trying to debunk this view that eating seafood, and it's repeated again in the press, even by science writers. They're not getting to the real issue is that there's no evidence. And not only that, but they completely, I've even got stuck in a very contentious study where I didn't know that the lab that I associated with was funded to find methylmercury toxicity in humans from consuming seafood, and I didn't know that going in. And so if you Google us, you'll see that in the Washington Post, we had a 14-year archive of bluefin tuna tissue that I wanted to basically go on record of methylmercury and selenium concentrations, just so the work was done. So the investigator only looked at methylmercury and said that,
didn't want to look at the selenium.
By the way, my selenium values aren't usable. And it was an unbelievable, and I had no idea.
It must be so frustrating to see science manipulated for political reasons like yours.
I just didn't know that it's not just a lab, it was the EPA versus the FDA versus NIH and NOAA. So it was a nightmare. But finally,
who was taking which side of these organizations?
Well, they set their levels, and that was EPA and FDA had they set certain levels. I don't know if it's at 1 part per million now, I forget. Whereas NIH, and who studied soldiers, who basically had much greater demographic information, they were saying that no, this is not a problem. But it's still going on. And so recently the press did cover the Hilbaum et al. Study, but they still had caveats, but maybe you shouldn't eat tuna, bluefin. And I thought.
Just maybe don't, for who knows why.
So chefs, it's not their job to know all of this sort of complex. I mean, it's complex.
So if there was a chef listening right now who has made that decision, they run a seafood restaurant, they're not buying or serving bluefin, what would you say to them, kind of, in summary?
I would say details matter, and see what your source of your bluefin is. Was it from the Atlantic and fished sustainably? If it was, I'd say serve it like Seattle. I met an amazing sushi chef in Seattle, and we sat down. I was there early. He and I sat down. It was early in the night, and he couldn't believe it. And he said, "You've made my day. You've made my month. I love bluefin. I didn't know I could buy Atlantic bluefin and feel good about it because it's on the Seafood Watch."
Yeah.
And I said, "Yes, you can, and you have that from not just me, my opinion, but you could look at how the U.S. manages.
Yeah.
The Pacific bluefin, that's subjective. I'd say make your choices. And I would never have eaten a Mediterranean bluefin and asked Charlie if a local restaurant was serving med fish back before they were coming into scientific compliance. I would boycott it. I would not eat it.
Before the fishery was in scientific compliance.
Before the EU members basically did the right thing.
And how they regulate, though, the fishery you're talking about.
Yeah, and they were in violation until about 2000.
Okay.
I can't remember now, like 2010.
Yeah.
But when I saw bluefin on the menu here and knew it wasn't coming from the American fleet, I wouldn't eat it.
Okay.
And I would talk to the chef. I would say, please don't buy that. And that's why even now, what I don't understand is, and I think you do, but why don't we have more distribution of really beautiful, when the Billy Baker story about when the price fell to $1 or $2 a pound, it fell for various reasons, particularly biological, but why don't we have distribution chains for bluefin? Why should they all have to go to Japan if they,
if I had known bluefin was $1 a pound, I'd have been buying whole bluefin. You know what I'm saying?
People were actually. I mean, I didn't even know about it. I just come back from Kauai working on yellowfin, but we kind of missed it. But there's the distribution chain to restaurants, and also cutting up the fish didn't exist for bluefin because of all these perceived challenges, overfished, mercury, all this stuff.
Yeah.
So that's a future. I mean, I think when you talk about local and sustainable, there's nothing more local and sustainable right now than bluefin. Not to say you should eat, too much of anything, but I'm just saying that the politics and sort of the challenge of the science and everything else meant that bluefin went to the exclusive high-end market in Japan, and now, of course, it's going to a much broader area, but it's all so challenging, and like any other commodity, it's a very interesting field, but it comes back to the Hillbell et al. Study found that not only did children that had high levels of fatty seafood in their diet, which would be the fatty seafood and the mackerels, the tunas, their cognitive performance was higher. Their methylmercury levels and their cognitive performance were higher than children that didn't have those kinds of seafood in their diet.
So they did have higher methylmercury?
They did have higher methylmercury.
But they had higher cognitive
and that's why it's such an amazing study.
That you get this perceived contradiction there.
They didn't mention selenium.
Yeah.
They didn't have to. They just looked at cognitive performance. They looked at all kinds of stuff. It was a major study, like big news. It hasn't made its way through yet. It hasn't hit the
filtered into the mainstream yet,
or the chefs and restaurants.
Okay, yeah.
But what I know from Joe Hillbell, back when he gave me guidance when we were having all this trouble with basically famous labs, it said, "Oh no, that's a myth that selenium is doing this." Wait a minute.
Because I've looked at this and I see both. Some stuff I read, yes, selenium's a great buffer against methylmercury. Other things saying no, that's not true at all.
They won't even cite the guy that the individual. He didn't even want to cite the selenium papers. He refused to cite them in our study. I said, "Well, that's just cherry picking. That's wrong."
Again, that's not science.
But coming back to what they found, because the NIH studies soldiers, that's why tuna is given as a what would you call it, the MREs.
Oh yeah, okay.
They
you're saying as for military?
Tuna that military relies on canned tuna, albacore, and other things. And in fact, the big canneries, they developed this foil pouches for the military. They wanted high quality fillets. And I didn't know any of this, but they actually study diet in military and try to keep the service members. So Hillbell not only looked at just the general population, but they're studying populations around the world and food and health, and even infants, because he's an MD, a PhD, and so he's one of the top experts with no real agenda. So it opened my eyes to, holy crap, did I ever step into it?
Yeah. I just have a couple more questions for you. For people who want to learn more about bluefin outside of academia, are there any popular books, documentaries, anything like that you think are worth looking at, especially if people just want to learn more about the general sort of life history, natural history of the fish?
That's a good question. I really love, if you're really serious about bluefin, the Frank Mather historical document, I know it's academic, you can get it from National Marine Fisheries Service, but basically Mather just consolidated, and his co-authors, the most basic biology of bluefin around the Atlantic, and it's excellent. I call it the Bible. All my grad students had to have it. As far as popular, there's a lot of misportrayal of
that's
bluefin.
what I'm now realizing after this conversation.
The 4 fish, I mean, if you go back, the only spawning in the Gulf of Mexico, only old fish.
You're talking about Paul's book, Paul Greenberg.
I think just they didn't have the time to understand.
Because we interviewed him. He's a fantastic guy. But
absolutely.
Not a biologist, obviously, or an ichthyologist, not an oceanographer.
I'm a little rusty on what he said, and he may have interviewed me, but
I think he's under the standard narrative, though, about
standard
these
narrative,
fish, yeah.
And you're right, and you can't blame them. But I mean, even National Geographic, I was interviewed where, and as I pointed out, that's why I know that even with sitting down with a scientist that can put peer-reviewed papers on the table with it's not just Molly. There's a lot of co-authors in all of our work with very well thought of scientists. Everyone says Molly because I've been the token sort of what's the word? I'm portrayed as a lone wolf, even in the larval papers. Even though the heads of the studies were all NOAA scientists, it was Lutcavage that they went after as far as, didn't even identify that there are many, many other scientists in our studies. And not only that, but National Marine Fisheries scientists to boot, as far as the larval, spawning grounds. So coming back to, I hope to write a book someday about some of this.
I hope that you do.
But
set the record straight a little bit,
I mean, there are certainly. I'll have to think about that.
Yeah, no worries. Another question.
Giant bluefin. Sorry, I take it back. It's
by Wynot?
Doug Wynot.
Okay, which I read that.
He
actually.
ended it.
Yeah.
The thing is, I tried to talk Doug into writing another book. He ended where we started.
Okay.
So what he will call the climax of his book is when the 2 spotter pilots, Jonathan Mayhew, when I was not allowed to present at the ICAP meeting in D.C. or Silver Spring, and the 2 pilots pulled their 3 minutes apiece and presented. That's where Doug's story ends. But Doug brought in both the biology, the fishermen, Mather, and the beginning. And I thought that was then just the beginning of the ENGO and policy adopting bluefin. That's where Doug ended. Yeah. And somebody needs to
pick up where he left off.
And the manner in the journalistic manner. And some people said, it was kind of boring. But you know what? I
really enjoyed that book, actually. He's sort of following along, especially with the harpooners, right? Out of here,
yeah. And I worked with most of those guys. But somebody needs to follow up. And the politics only got murkier. And I will say that Frontline... there was going to be a Frontline documentary on bluefin and the politics of bluefin, by the very famous head of Frontline documentaries. What's his name? David Fanning. But he was in my kitchen when he got the call from National Geographic that they were pulling the funding.
They don't want to touch it, huh?
I don't know why, but ask Charlie. He was asking me for setups to meet other fishermen for the documentary. Right. But he was going to cover the politics.
Oh, it's so interesting. It's just so complicated. What's next for you in your work, and or in addition, not just what's next in your work, but what do you hope is next in the bluefin science?
Well, it's an exciting time for bluefin science because you could probably tell I like the edge. I like the envelope. I don't like the cleanup operations. Mm-hmm. I like the frontier, and I think my colleagues do too. We like to go after the big holes, the mysteries that maybe other people don't care about, but we do. And I'm excited about bluefin because now that we know that bluefin are spawning across a big area, now that we know that there are a lot of unaccounted for bluefin, the really exciting thing is what does it all mean? Because what we didn't get into is, this whole assumption of 2 stocks and natal homing. Genomics and genetics is going to tell us if you sample far enough, are we cousins? Are you my grandmother or grandfather? For the fish that are looked at, and I think what we're going to find out is now that we know from tagging done by a lot of groups that there's so much more mixing going on than we ever dreamed, and that there's switches, so that you might do the same thing for 3 years, or we don't even know now that there's only 1 spawning ground per season. The Japanese found some separation, so we have tracks of fish that are sort of in the Bahamas in warm water in February and March, and then they'll beeline into the Mediterranean for June, the known spawning period, and then they're back up off southwest Nova Scotia in November, where we put the tag on.
Wow.
Not most of them, but is it possible that the Atlantic population is what the old guys said it was, one big population with tribes, or what we would call subgroups, or a metapopulation?
Yeah.
That's where I would put my money. I don't think the two, spawning site fidelity, I don't think makes sense based on tagging results and ecology and life history. There may be trends and patterns. And then finally, what we don't know is how tuna navigate, how they share knowledge, and basically how do they get around. When we had one bluefin that we tagged, caught it on an anchor off Southwest Nova, tag stayed on a year. During the year, the fish ran over to the Azores in March, fed there, wasn't spawning, it was too cold, probably diving on deep diving on the squids, just like the whales, came running back over. A year later, the fish was 20 miles from where we caught it. Even though I'd run across the line, 20 miles, okay, 20 miles, pretty close. 2 weeks later, that tagged fish was caught by a fisherman in the Southwest Nova group, Garth Nickerson. It still had the pop-up tag tether that I had made and that attached the pop-up tag to the fish. It was caught on the same anchor that we had caught the fish on.
Came all the way back.
All the way back after going to the Azores.
Wow.
And to me, that wasn't by chance.
Yeah.
So we know nothing about how
they
all of these
navigate,
big animals and whales, sharks, we know there's magnetite in the brain of everything. But so the real scientific frontiers are, how do they do it?
Yeah.
And what's the relatedness, and et cetera, et cetera. Do you think stock assessment scientists cares about this? Not too much.
Right.
That's on the edge of science.
Right. What about your legacy? I would say it's become clear to me looking over your work that you were the front person for so much of this. Like you're the one out there. So I
wasn't alone.
You absorbed a lot of the arrows, right?
Was it because I was a woman? I hate to bring in sexism.
What is it, right? You sucked up a lot of the arrows, right? Like you were saying the things, or you were perceived as the one. So, but historically, the way those things usually go is you'll be the one remembered later for having done the really important work when no one would listen, right?
Oh, one hopes, but...
What do you hope, outside of all of that, like what do you hope the legacy of your work is
I hope
forward?
Just that, and I just hope, I hope we shifted the paradigm that working with fishermen is a good thing for science.
Yeah.
And we didn't start that. The old-timers taught me that, and... I don't know if I have a legacy. I joke I'll be
you definitely do.
Unemployed. And how could we be unemployed and without a lab? That's what Billy said, his editor said, are you sure she's not just a jerk and a bad scientist that she lost her lab? And that was a fair question. But
I wondered that on my way down here.
I sent him my CV in all our publications. Well, hey, we're published in the top journals, and we were right. We're still right about a lot of this stuff. So, no, it's just politics and money and, just timing and... But I'm not done, and I love working in the Pacific with fishermen. The Pacific is still so much to do in the Hawaiian Islands and, the territories, and the enthusiasm there is the same as for here. But I hope that fishermen will feel that that scientists aren't the enemy, and I hope they will continue to care and to push for better science and not just accept status quo because it's their business. And as a scientist, if you're not honest about doing the best science you can and doing your job and basically supporting your neighbors, then you're not a good scientist. And it's taxpayers' money that we spent doing the work we did. Right. So it's my job to get it right. And even though it's not particularly my job anymore, because we have a hard time seeing these views are not easy to fund lately, and you need funding. So. My legacy is we'll continue to work as long as we can and to continue to associate with the best scientists that we can work with and to publish.
Wow. Thank you so much for sitting down with us today, and thank you for all the work you've done. And on a personal level, I'm going to go have some bluefin today. So I want to say thank you because it'll be the first time in a while I've eaten it with feeling good about it. So.
Oh, bravo. Well, I really appreciate the time and your caring to hear all of it. And I appreciate what you do. And as I say, you have a lot of friends around the world that want to do the same. And you should come out to Hawaii, and you would fit right in.
Yes, please.
And so we'll talk to you in Hawaii, I hope, too. Or come back, and hopefully we'll have more news for you next time. If we ever talk on bluefin again, I'd be thrilled to tell you what we learned the last couple of years.
If anybody wants to follow up on your writing or your papers, anything like that, we'll put notes in the show. But do you have social media or websites or anything like that?
We do. I mean, you know my age, so you know I'm not probably as good as I should be.
You're not on Snapchat.
I'm not on Snapchat, but we do try to host, and we try to focus on the science by putting it up on Facebook, like links for people that want to follow up. So our website is tunalab.org.
Okay.
And it's a bit static lately.
But you give talks around here and stuff too, don't you?
Yeah, and we've had over the years a series, a public seminar series, Fish and Fisheries, that we hosted. And so tunalab.org, and then our Facebook page is LPRC, Facebook LPRC Tuna Lab. And that's where we put our current stuff up. And we're a little behind because there are only two of us right now.
That's life.
So
people understand that.
Yeah, I need to find an 18-year-old too. Yeah. I'm sorry I'm being ageist, but somebody to...
can be ageist with us, we don't mind.
And the Tag a Tiny program, that's citizen science. All our recreational fishermen that want to tag and release are part of now 2,000 plus strong in the Atlantic, and in the Pacific it's fishtoday.org, fishermen that are tagging and releasing their
juvenile tuna. Juvenile bluefin,
juvenile or billfish, or even what we call the bottom seven, the snappers.
Oh, cool.
In Hawaiian Islands. So there are ways to engage in ocean science and get more out of your release by putting a tag on it and contribute to the scientific understanding.
Oh, that's cool. All right, great. So we'll put links to all that. Molly, thank you so much for your time today.
It's been a pleasure, Daniel, and enjoyed it. Thank you.
Thanks for listening to the WildFed Podcast. You can help us grow this show by subscribing and leaving us a rating and review. It ensures better rankings and more advertiser interest, which translates directly into better shows, more awesome guests, and a constant stream of fresh, new content. Have a question you'd like answered on the show, or a hunting, fishing, or foraging trip you'd like to host us on? Email us at info@wild-fed.com. And be sure to visit our website, wild-fed.com, to check out the WildFed video show and store. WildFed. Food is all around you.